Examining variations in mineral chemistry can shed light on changes in volcanic activity and allow researchers to obtain timescales of volcanic processes to better understand any potential hazards.
Atom probe tomography allows an unprecedented 3D visualization of mineral exosolved phases, while measuring their chemical composition at atomic scale. We are currently applying the technique to the characterization of pathological mineralization, for example, in the case of kidney stones.
This method can be applied to volcanic systems, where eruption transitions can occur over small timescales. These transitions are recorded in the minerals over very small spatial scales.
Begin by pouring 1 gram of the sample into a 10 centimeter glass petri dish and wrap a 3 by 3 centimeter sheet of weight paper around a 10 Gauss magnet. Use the magnet to pull magnetite-rich grains between 100 and 500 micrometers in diameter from the ash sample, and place the grains in a 32 micrometer pore, 8 centimeter diameter stainless steel sieve. Use a squeeze bottle of deionized water to flush the smaller adhering ash particles through the sieve for 20 to 30 seconds and allow the grains to air dry for 24 hours. The next day, affix any clean and dry ash particles to sample mounts suitable for secondary scanning electron microscope and image the particles in secondary electron mode at a 15 to 20 kilovolt accelerating voltage and at a working distance of 10 millimeters to select the 5 to 10 best candidates for further analysis. The selected grains should be predominantly magnetite. Affix the selected ash grains to a piece of clear tape and surround the samples with a 1-inch diameter hollow mold that has been internally coated with vacuum grease.